Today we'll talk about DNA replication and enzymes. To begin with, I'll talk about DNA replication. Most enzymes end in ASE, however, some do not. Names of enzymes that end in ASE is like such as catalase, invertase, and carbonic anhydrase. Catalase removes toxic H2O2 from cell. Invertase breakdown of sugar. Carbonic anhydrase hydrates CO2 gas for transport. Names of enzymes that do not end in ASE, such as pepsin, trypsin, chymotrypsin. Pepsin, digestion of proteins in stomach. Trypsin, further digestion of proteins in small intestines. Chymotrypsin catalyzes the hydrolysis of proteins, degrading them into smaller molecules called peptides. Secondly, enzymes are helpful in DNA replication and protein synthesis. They help by breaking the hydrogen bonds between the bases and allowing the strand to duplicate itself. Some reasons for DNA replication. First, for cellular reproduction, growth/repair of an organism, adaptation due to genetic mutation. During DNA replication, mutations can occur. Mutations that occur include missense mutation, nonsense mutation, deletion, and insertion. Missense mutation results in the substitution of one amino acid for another in the protein made by a gene. Nonsense mutation, a single nucleotide is substituted but leads to the formation of a stop codon in mRNA at the beginning of the gene, hence no protein will be produced. Deletion, in this type of mutation, only one nucleotide is deleted, and this will lead to frame shift mutation. Insertion, the addition of a nucleotide in the DNA. This is the video. No. ما أعرف بس هذا. Now I'll introduce you to Basil Jbeir. He will talk about his poster.
Okay. Assalamu alaikum. Today I'm going to talk about mutations and how do mutations occur. Every time a cell divides, its entire DNA is duplicated, so each of the two resulting cells have full set of DNA. Mutations can occur in a number of ways, such as normal duplication errors, or it can be an change in an environmental, such as sunlight, UV light, etc. Our cells have a mechanism that can repair the DNA, mutations. But the older we get, the less the efficient, the less the older we get, the mechanism efficiency gets weaker. What kind of mutations are there? A gene is made up of 4 nucleotides, A, T, G, C, that describe how to make a protein. Any say?
Today I'm here to talk to you guys about DNA synthesis and more specifically about blending strands.
Any changes? Any change in nucleotide alter the protein that is made. I'm going to name 4 types of mutations. We're going to use the big fat cat ate the rat as a sample gene. First kind of mutation, missense mutation. It happens when one single nucleotide is replaced by another and the protein produced will have no function. Example, the big fat cat ate the rat. After missense, the zig cat ate the zig fat cat ate the rat. As you can see, it doesn't have meaning anymore. The second type of mutation is nonsense mutation. The nucleotide is also substituted here, but instead of having a protein with no function, we get a stop codon, and hence no protein will be produced. Example: UUG, CCU, AUA, UAC, CAU, GCC. This is a normal gene and it produces a normal protein. But after nonsense, the UUG will change to AUG. This will make this will make the gene nonsense. It doesn't have a meaning. The third type is deletion mutation. Only one nucleotide is deleted. This will lead to frame shift. Example: the big fat cat ate the rat. After deletion, the sentence doesn't have a meaning. The big ate a rat. Fourth kind of mutation, insertion. An extra nucleotide in the DNA. Example: the big fat cat ate the rat. After insertion, the big fat doesn't have a meaning at all. The sentence. And now what Professor Sayas is going to present his brochure.
Assalamu alaikum. We all start off with mutations. Some are harmful and some are useful. But the most memorable mutations are the ones that cause diseases. In this brochure, I will list some mutations. Some diseases caused by mutations and their different names, their symptoms, and the people afflicted with them. And what kind of medical assistance does these people need? And end with the question that is a roadblock for many scientists nowadays, and that is curing mutation diseases, and how to introduce a gene into millions of cells of which carry the deflected gene. In the first page, you will see an introduction about mutations. Then you will see cystic fibrosis, which is a recessive genetic condition caused by faulty gene. Then you will see what symptoms does cystic fibrosis show, and what kind of medical assistance does people afflicted with it need. In the end, it will show you what kind of help you will need. This is our science project made by Professor Sayas, Mohammed Al Yahya, Basel Jubair,